So what we're doing today in this video, we'll discuss about domain Archea. Okay. So first of all, let me tell you the domain Archea. So this Archea is basically the diverse group, and initially this domain Archea it was known as Archaeobacteria. Okay. Initially the bacteria and Archaea were in the same group, so that's why it was known as Archaeobacteria. So let me tell you, Archaea is a diverse group. Initially it was known as domain Archaea. Oh, sorry, Archaeobacteria. Okay, initially it was known as Archaeobacteria. Then now they are placed in the separate domain. Like Archaea have another domain, however, bacteria have another domain. So they are separated. We can say they have now separate domain. And because why they have separate domain? Because it was proved that Archaeobacteria are slightly different than we can say bacteria. Means Archaea was different than bacteria. Different than bacteria. Okay, it was slightly different than bacteria. Then now let me tell you what is the diameter of this archaea. So we can say the diameter ranges from we can say 0.1 to 15 micrometer. Remember that this question is asked many times. Okay, what is diameter? So this is the approximately or ranking diameter. It will rank from 0.1 to 15 micrometer. Okay. So if you talk about the shape of this domain archaea, so domain archaea have different shapes like it can be in the square, lobes, spherical, or we can say rod. So the domain archaea have multiple we can say shapes, and remember this domain archaea do not contain peptidoglycan in their cell wall, do not contain peptidoglycan. However, it have pseudopeptidoglycan, means both it false or you can say true no, sorry false peptidoglycan होता है, which only help in the antibiotic resistance, जो कि सिर्फ एंटीबायोटिक रेजिस्टेंस के अंदर ही हेल्प करता है. तो do not contain peptidoglycan in their cell wall. They do not contain peptidoglycan in their cell wall, okay, but they have we can say pseudopeptidoglycan. Pseudopeptidoglycan which is present or which help in the antibiotic resistance. Antibiotic resistance, okay. I hope this is clear. Now let me tell you they can reproduce. How we can say they reproduce, we can say domain archaea. So they reproduce through binary fission, through fragmentation, and we can say budding. Reproduce through binary fission, fragmentation, likewise budding. Okay, so yeah, from these three, binary fission, fragmentation, and budding, they reproduce. Then now we have they have we can say different. They are different from bacteria in some metabolic activities. So let's see how they are different and why they were placed in in another kingdom. So they are different than bacteria. So number one was the condition they are very extremophiles. Like they are also known as extremophiles because they can survive they survive in extreme. However, they live in the moderate. वो moderate के अंदर रहते हैं लेकिन वो बहुत ही extreme condition के अंदर भी survive करते हैं. So they are called as or also known as extremophiles. Extremophiles. Now here we can say for the extremophiles we have three types. Number one we have thermophils. Another we have acidophils, which are for the acid. And third we have halophils. Okay, so if I to talk about these thermophils, so these thermophils are basically they can survive in hot spring, as the thermo word means heat or the condition which is very hot. So they can survive in the hot spring. So because and they have thermo ability stability which is around 120 degrees Celsius. They survive in hot spring. They have ability of thermostability. Thermostability and which is around 120 degrees Celsius. Okay, and if I talk about these acidophils, so these acidophils can live in highly acidic condition and which is around 1 to 2 pH, and you know 1 to 2 pH is very high and acidic. Okay, so they can survive, can live in highly acidic condition which is around 1 to 2 pH. Okay, and if I talk about halophils, so these halophils are which live in salt conditions. As halo we have studied in we can say metric class, which was chapter thermoregulatory like this. Okay, so here we have they live in high salt condition concentration. Okay, now here we have another we can say further, which is methano. We can say methanogens. Now methanogens are what? Methanogens are also which are found in marshy areas. Here we have methanogens. Now methanogens are we can say this is also one type of characteristics in domain Archaea and they are found in marshy areas. And we can say they produce methane gas and they help in the digestion too. Produce methane gas. And this question can be asked, what is the characteristic in which we can say produce the methane gas? So your answer must be methanogens. Okay, and methane gas, you know, it is the biogas. Okay, I hope that it is clear. After that, here we have let's discuss about we can say composition of the cell membrane in domain Archaea. Okay, so the plasma membrane of Archaea contain basically lipids that allow the functions of high temperature. As we have discussed, these were extremophils, and you know that lipid of we can say Archaea contain glycerol, and they are linked to branch chain of hydrocarbons. Lipids of this Archaea contain glycerol and chain of branch chain of hydrocarbons. Okay, and in the comparison of bacteria, the bacteria composition is like that. In the bacteria, it contain basically glycerol and linked to the fatty acid chain. Glycerol linked fatty acid. So that is the in the we can say bacteria. Okay, I hope this is clear. Now here let me tell you about the we can say ribosomal sequence. Okay, so remember that the ribosomal RNA sequence. Mm, okay, so here remember the most fundamental which is different between Archaea and eubacteria. Like you might be thinking as Archaea have composition which is glycerol plus we can say long chain of or branch chain of hydrocarbons. This is same like eukarya have. So now what's the difference between we can say Archaea and eubacteria? So they have basically in their nucleic acid they have difference in the nucleic acid. Nucleic acid. Now how they have difference? Okay, let us study, okay, they have difference in their nucleic acid, like how, for example, their nucleotide number is 91 out of 1500 is one type of RNA which is founding differences. Like we have 91 out of 1500 we can say nucleoid numbers. So in this condition, the we can say one difference can occur. Like for example, if you bacteria have the sequence of A A, C, U, C, A, and A. However, at this, the Archaea have the sequence. Archaea have the sequence which is A, C, U, C, A. So here you can see there is only the little bit difference between the Archaea and eubacteria. Here this is same, this is same, this is same, these are both are same. However, here we have C, which is cytosine, and here we have U, which is uracil. Okay, here we have uracil. However, here we have cytosine. So this is only the difference between Archaea and eubacteria. Okay. So I hope this is clear to you all. Now in the next video we're gonna discuss about bacterial ecology and diversity. Till that, bye bye.